Hi, it's Paul. I want to look at respiratory specimens and the different cell types you see in the different specimen types. They vary because the areas of the lung they sample are different. So we'll look at sputum first. This is exfoliative cytology, so the cells have to fall off and be coughed up. And so what we often see is a predominance of oropharyngeal material, that'll be squamous epithelium, and mucus and debris. This has got quite a lot of macrophages, just the one squamous cell pretty much in this field. This is another more typical field probably that has a lot of squamous cells, a lot of debris, and then scattered around are alveolar macrophages. We use these as an indicator of adequacy for sputum specimens because these guys live in the alveoli, and if they're present in the sample, we at least know that it's been a deep cough and the whole airway has been sampled. They typically have a histiocytic appearance and then pigment in them, as you can see, especially in that lower one. A couple more over there. Also, you see in sputum a lot of junk, really, and this is a whole lot of debris out of the oropharynx. Sometimes all you get is saliva, and it'll sometimes have the patient's breakfast in it. So these are plant cells. This is another big sheet of plant cells, and you can see they have quite thick cellulose walls to them. They have nuclei that don't have a chromatin pattern, and these vary enormously in appearance. Sometimes that group, they can look a bit like squamous or squamous metaplastic cells. Here's a few more here that don't seem to have a nucleus. And to go with that, you often will see this. Guess what this is? So this is striated muscle. So this is meat that has been eaten. You can see the little striations, and so it looks just like skeletal muscle from a human. So that's the meat to go with the veg. These are a really nice example of some alveolar macrophages that have anthracotic pigment in them. This is carbon. Here's a nice field with lots of macrophages, some squamous cells, and this wiggly material is called a Kirschmann spiral. It's just inspissated mucus, so it's like a cast of the very fine airways. It's non-specific, it's not reported, but it's seen probably more commonly in people with very mucoid conditions like asthma. Good macrophages here showing pigment. You'll see how variable it is. So these guys have, typical histiocytic morphology, so round cells, foamy cytoplasm, often with ingested material, a nucleus that's often eccentric. The other thing you see in sputa sometimes are bronchial epithelial cells like this. They're quite a tall columnar cell, as you can see. You don't always see cilia, particularly in sputa. They're fairly fragile. I can see it on that one cell there. Often what you'll just see is like a terminal bar where the cilia would meet the cell. So these are not terribly common in sputum, but very common in bronchial washing specimens. And that's what we'll look at now. Brushing specimens tend to be prepared in theatre. So these are often very bloody, so they're fine sampling of the lesion, as I'll show you in the lecture, with a tiny brush. So the cells are directly removed. You have usually quite a bit of blood as a result. These are spread onto a slide, usually by non-laboratory staff in the theatre, and so what you often see is a poorly spread slide, lots of air drying, and they can be a little difficult to interpret in that way. We usually wash out the brush as well, or the brush is sent, I should say, in saline, and we then wash it into that and often get a lot better morphology from that. Washings are performed at the same time because they're already there, and often the morphology is better because they're sent as a fluid and prepared in the laboratory. So here's a typical group of bronchial epithelial cells. You can see the little red cilia even at this power on them. These are just like a little crew cut of red hairs running along the top. Notice there's a darker part at the base where it meets the cell there. That's the terminal bar. Often the cilia are lost and you'll just have that terminal bar present. Notice they're quite long columnar cells and they're really quite uniform. Those there are goblet cells. You'll see them occasionally. It's hard to see the morphology, but you can see the clearing of the goblet of the mucin. So in bronchoalveolar lavage, or BAL, what we're doing is sampling the alveolar spaces. So you should have predominance of alveolar macrophages, which is what we've got here. Often there's a bit of contaminant, so you can see squamous cells around here, and you'll sometimes have bronchial epithelial cells scattered in amongst those macrophages as well. But it should be pretty much a deep sample of the lung airways. Just one final thing, these are a non-cellular finding that we see sometimes in patients who've been exposed to asbestos, and these are ferruginous bodies. So they are asbestos fibres that have had iron laid down on them, and they take up this kind of dumbbell. These are found in patients who've been exposed to asbestos. If you use an iron stain on these, then they light up quite strongly as well. But this is a typical appearance under Pap stain. And so in summary, in sputum we're sampling the upper airways, so we can have quite a bit of oropharyngeal contamination. Squamous cells, macrophages indicate that it's adequate, and you'll see rare bronchial epithelial cells. In bronchial brushings, you'll be directly sampling the lesion, and so you'll see bronchial epithelial cells and maybe tumour cells if it's a malignancy. In washings, you once again are getting upper respiratory contamination, so they can look quite like sputum, but they generally have a larger component of bronchial epithelial cells. In BALs, we've got a deep sample, and these should be predominantly made up of alveolar macrophages. You will occasionally get some contamination with bronchial epithelial cells and with squamous cells, but they should be ideally a pure sample of the alveolar macrophages and some